Welcome, everybody. Let's go ahead and get started. Our speaker today is Todd Hollon. He's a neurosurgeon and research scientist who specializes in brain tumors. He is an assistant professor of neurosurgery, and he did his postdoctoral training here in the Translational Molecular Imaging Laboratory under the supervision of Doctors Daniel Orringer and Honglak Lee. His postdoctoral work focused on the application of deep neural networks to advanced imaging methods to improve the speed and accuracy of intraoperative brain tumor diagnosis, which I believe he'll tell us more about today. And he has recently joined CCMB as an affiliate faculty member. So we're excited to hear about what he's doing. So Todd, I'll turn it over to you. Thanks for speaking to us, and we're excited for your talk.
All right, thank you everyone for being here. As things get more and more in person, it's good to see everyone out. So so I'm a neurosurgeon, and my work really focuses on how we can use techniques from computer science, artificial intelligence, and bioinformatics to improve the way we operate on brain tumors, both in terms of how we improve the way we diagnose brain tumors and also how we treat them. And those two are obviously related. So this talk is really about how I have been applying a novel imaging technique to a specific set of artificial intelligence methods in order to improve brain tumor diagnosis, and particularly brain tumor diagnosis in the operating room, which is where I spend a lot of my time. So there we go. Okay, these are my disclosures. I am a shareholder in one of the companies that makes the imagers, but unfortunately, I haven't seen any benefit from that yet. So, yeah, exactly. So this is really the problem that I'm faced with daily as a clinician, is that we see these brain tumor patients and they come to us after they have a seizure or a headache or a new neurologic deficit, and all these scans look about like this, right? There's a lesion in the brain, contrast enhancing. The radiologists tell us what it looks like, rim enhancing with surrounding cerebral edema, and they just describe what this is. But ultimately, this does not give you a diagnosis. And the only way to get a diagnosis is to obtain tissue from these tumors. And the problem that we face is that there's lots of different ways for us to get tissue. You can do a small biopsy, you can do a large resection, you can do a subtotal resection, lots of different things that we need to take into account to make sure we don't hurt the patient, but we also give them the best possible therapy we can. If that was the only problem, it wouldn't be a big deal. But the problem is that diagnosis actually determines the treatment, right? So if you have someone that has a tumor that's very responsive to chemotherapy, radiation, you don't want to do a big surgery. You're going to hurt the person, right? Or, for example, if it's multiple sclerosis and you see it and it looks a lot like this, which they can, you want to do a biopsy and then get the diagnosis and back out. You don't want to do anything at all, because that's a whole different set of biologics and anti-inflammatory medications. So we run into this loop problem where the best treatment is defined by the diagnosis, but we don't have the diagnosis yet so we don't know what the treatment is. So this is the issue that we face daily. And this is just to show you, this is what my world looks like a lot of days. So this is the brain. This is a patient I did last week, and I do a lot of awake brain tumor surgery. And this patient is awake and talking to us. And we've done our mapping. We want to find out where all the most important structures are, like language and motor function. And we've done this, and you can imagine we still don't know. Is this a multiple sclerosis lesion? Is this glioblastoma? Is this lymphoma? All of which have different treatment options. So, yes, please.
You do stereotaxis to get inside. I mean, that's the surface, but you got to get inside a lot of those. So
Exactly. We do, yeah. So we have frameless stereotactic navigation that we use, yeah. And that's an important part for localizing where we should best enter the lesion. Unfortunately, still it comes up short in terms of what we're actually dealing with. But we do use navigation for all of our craniotomies. And please stop me at any time. I would love for this to be interactive. So we have developed a solution, but it's a bad one. I don't think there's any pathologists in the room, but I think they'll agree with me on this. So this was a paper that I found. Harvey Cushing is like the person who invented neurosurgery back in the turn of the century. And he defined a method that was intraoperative diagnosis, where you take a little piece of tissue, you do some staining on it, and then from that, a pathologist looks at that and tells us what they think the best approximate diagnosis could be. Now, this is not a final diagnosis. This is a tentative sort of best guess that you can get from very rudimentary analysis just from using light microscopy and some simple staining like hematoxylin and eosin staining. So the images you can see on the left look a lot like the images on the right, and these are images that came, from 2 years ago or something. And there's a lot of problems with this. It's really a quick and dirty method where we freeze it and then we section it and we stain it, and all of which introduce artifacts, which makes the diagnosis that much harder, specifically for the pathologist, the humans, the reading this. So this really sets up, this is the state of the art. This is the standard of care that we have, which there's clear limitations. So the question is, can we develop a streamlined intraoperative pathology workflow that is fast, efficient, and accurate? And nobody has been able to do this constructively for the past 100 years since it was initially invented. So this is really the overview slide of the whole research program that I have been a part of. The top arc is the normal standard of care, where we have a conventional pathology lab that usually lives on site somewhere near the operating rooms, but sometimes it's off site. Where I did my fellowship, it was in a completely different hospital, which made it completely infeasible. We do H&E histology, and then there's a neuropathologist who's on call, which they do not enjoy. And they have to read these. They have to be called out of their lab. They're doing their animal experiments, and they have to come in and, you get their page and they say, you have to come read this intraoperative frozen pathology specimen, which they don't love, all of which takes anywhere from a half an hour to 2 hours. And then the lower arc is what we're working on. So we're trying to replace each one of these stages. And the first bit is the imaging. And what I've been a part of is developing stimulated Raman scattering microscopy and the translation of this imaging technology from what was optics labs into now the operating room. We're developing how to process these images, and we've developed a system called stimulated Raman histology. And then where my really scientific work has come in terms of application of AI algorithms, specifically computer vision, trying to translate the latest stuff that's published on arXiv and all these amazing techniques that have come out really the last 10 years. We're trying to leverage that to do the best that we can for really intraoperative decision support, automated diagnosis or augmented diagnosis, however you want to call it. And it gives us an order of magnitude faster time for us to get this diagnosis back. So it's been a long haul. This isn't even the start of it, but we started in this is an image on the upper left of an optics lab at Harvard, where this technology, the imaging technology, really started. And my mentor, Dan Orringer, actually was really instrumental in translating this into it being a clinical tool. Our initial prototype imager is in the middle, and then what we're using now actively, what is actually a commercially available system from Invenio, is this Neo imaging system, which we now use regularly in our operating room. So I use this today. I had an operation this morning, and we were using this to image a pituitary specimen. So it's really been a true kind of translational science. It's been a wonderful thing to be part of something where it actually goes from the bench top, and now we're actually have a clinical tool, and we're doing research with the data that's being generated from this imager. So this was the original paper, 2008, published in Science. So Chris Freudiger, the lead author, is actually the main R&D person at the company that builds the microscopes, and Sunny Shi is a very seasoned optics investigator. And this is the idea. So just a quick overview. So spontaneous Raman spectroscopy, maybe we did in organic chemistry in undergrad, but really it's just shining a laser beam at something, and you actually just collect the scattered photons and you look at the spectra that's generated from the media. And that spectrum is determined by the actual biochemical properties of the tissue. So you may say infrared, you do this. If you have a purified substance, you're trying to determine what that substance is. Similar thing can be applied to biological specimens, and that's what we do. The problem with that, and people have tried to do conventional Raman spectroscopy, is it's really slow. There's a lot of averaging and the signal is very weak. So you really have to spend 30 minutes to an hour just imaging the same single spatial position in order to get the spectra, which you see right here. And you get this nice spectral information, which has important biochemical information, but it just takes too long. This isn't a feasible thing for us to do in the operating room. So the idea with stimulated Raman scattering microscopy is rather than using a single laser beam, you actually use two. And by using two, if the difference in the wavelength between those two lasers is just right, you get this nonlinear resonance of specific chemical bonds in the tissue. So in this case, there's this, so the wave number 2845 and 2930, you get this narrow band nonlinear amplification of the signal, and you can generate an image from that. you can capture an image at this very narrow band wave number. And that's what gives you these images on the right-hand side. Now, this is from the original paper. I'm going to show you images that look a lot better than this, but this is how it all started. And we're still using this same technology in order to generate our images.
So that's where the word stimulated.
Exactly. Yeah, the stimulation is really from these two coherent lasers that are amplifying these Raman active chemical bonds.
And then that picks up on certain Raman, the Raman process picks up on chemical bond vibration.
That's exactly right.
Which ones are you picking up?
Yeah. So there's really two main frequencies. So we picked this strategically. These are normal biological specimens. So there's a lot of carbon. So it's actually the CH2 symmetric stretching mode, which is the main one. And then the other one is the CH3 resonance, which is really common in proteins, right? Methyl groups, right? So those are the two that we focus in on. And actually our images, I have some pictures of this. Here we go. Perfect. So the 2845 on the left is the CH2 symmetric stretching, and the 2930 is actually going to highlight mostly protein in the image, the methyl groups that we see. So we're basically getting we focused in on these because they highlight different specific things, even though, some bright pixels are bright in both images, but they seem to give us this excellent chemical contrast that we're looking for in terms of generating the images. And the thing that I'll point out that's different about this is something that's really hard to communicate to people. They say, why don't you just do H&E, right? There's a lot of H&E data, right? The problem with H&E is that the image contrast is completely artificial, right? It's a stain that we use that's just something that is easy to differentiate nuclei from cytoplasm, but it doesn't reflect the concentration of, let's say, lipid, like fatty acid tails, or if the concentration of protein or blood or whatever it may be. So, but this is in order to get that image, there's no staining, right? There's no special stain, there's no fluorescence, nothing.
It's not a visible light,
Right.
Down into the infrared somewhere.
Right.
Where's 2845, right?
Yeah, that's near infrared there. Yeah. So yeah, you got it. Yeah. So this is again where, it's like there's the advantage of this is that it's not stained. There's no processing that goes into this. I take a hunk of tissue out of the brain and I put it in a pre-made microscope and we put it in the imager and we get these images out. So our initial iteration was this blue-green color scheme, which I actually really like because it makes the images look cool and futuristic, but the pathologists don't like them so much. But the images on the right-hand side are actually the one-to-one correspondence between the tissue that we image and the H&E, which everyone knows and loves, right? And we basically see really good similarity between the two imaging modalities.
You got a lot of structures in there.
Definitely. I love it. I love that you point that out. This is great. I love it. So the advantage of this, you see how bright some of those structures are in the blue-green color scheme, the stimulated Raman scattering microscopy image. The reason for that is because we're actually imaging myelination in the corpus callosum, I think. So we're actually imaging these dense lipid-rich myelination that's happening. You don't get that in H&E. That's not something that H&E can handle because there's not something that's specific to either the hematoxylin or the eosin. So this is where you're actually reflecting back to you something important about the tissue itself, rather than just, this is more, basophilic versus acidophilic, right?
Well, there's something else going on, and those things are line fibers, so you're getting some kind of directionality effect in the scatter.
Right.
That you wouldn't pick up with an H&E.
Yeah.
Light
Right.
So for white matter, it's going to be phenomenal.
Right. Exactly. Yeah.
It's basically tunneling.
Exactly right. Yeah. We image definitely the white matter we image, and particularly white matter tracks look fantastic in this. That's one of the things that the pathologists really like, is that for these infiltrative tumors, it can be hard to differentiate intrinsic brain tumors like gliomas versus metastatic tumors. They both look very malignant, but if you see a lot of myelinated axons admixed with tumor, that means it's got to be an intrinsic tumor, right? It has to be something that's actually biologically part of the brain itself. So that's something that has been a real important difference in H&E that has driven a lot of adoption from the pathology side, is they see these advantages to having this label-free imaging that highlights specific things. So Yeah, there are limitations. So this is all ex vivo imaging. So we're taking a piece out and then we squash it into an imaging chamber. The imaging chamber thickness is about 120 microns. That can change a little bit, but it's imaging and transmission where you actually do get the reflected laser on the other side, the objective. There is an epi transmission mode where you're actually getting the reflectance back. It's technically more challenging, but it is doable. I think actually these images may be in epi mode, but point being is that it's an optical sectioning and you can set that. I think we have it set as default at like 20 microns, but that is a parameter you can adjust. Yeah.
So you got 120 micron section, you got 20 micron optical section. That's phenomenal.
Yeah.
So your resolution now is on the order of half.
Right, exactly right. We were just talking about this yesterday, actually, in lab meeting, what the exact resolution was, and it is. Yep.
It's pretty great. That's great. And so we'll get to the ultrasound.
Right. Yeah. So one of the things after we had shown that this images brain tumor specimens really well, we wanted to show, okay, you need to be able to do something with these images. You want some quantification. And this was right before I got into the lab, even though I was sort of involved with some of this, but, using really simple, what I think of as biomarkers. And in this case, it's exactly some of the things that we've talked about. How much myelin is in the image? How cellular is the image? What's the ratio between the CH2 and the CH3 channel? That's 2845 and 2930. Can we use that to make some predictions about the degree of tumor infiltration, which is ultimately what we are interested in as neurosurgeons? And sure enough, you can. I mean, these are simple, very simple classifiers, which are really just trying to say, what is the likelihood that there's tumor there or not, just based on really three features. So the three features were axonal density, nuclear density, and then protein lipid ratio. And from that, the bottom right is the classifier value. And again, this is just a binary predictor where you have 1 if there's tumor, 0 if not. And you can see we had really good results. So this was very preliminary. This was sort of the start of the introduction that had happened prior to me for the uses of computer vision for quantifying tumor infiltration. So I like to point this out because this is something that I think is confusing for some people who don't look at this from the perspective of bioinformatics. How do you make a diagnosis? Really, like what do you have to do to get there? And if you get it down to the most basic elements, I think there's 2 things. You have to have some data, some observation, right? Some measurement. That could be an MRI, could be sequencing data. It could be, it could be optical imaging. Whatever it may be, you need some data point. And the other bit is you need some analysis, right? That data doesn't come pre-interpreted, and that's what you need in addition to the raw observation. And the assumption is always that humans make that interpretation, right? Whether it's defining normal ranges for sodium levels, whether it's interpreting MRIs. And the point that I like to make is it doesn't need to be that way. There are other ways to make interpretations, and we can potentially assist humans in making those interpretations using a lot of the new AI or even old AI that's available to us now at our disposal for improving how we treat patients. So this is where, this is sort of the point of the talk is for a diagnostic system that I'm referring to in this case for both intraoperative diagnosis and even postoperative diagnosis, and even for molecular classification, which is what we're going after now. We're saying that you can do this with optical imaging, which has important quantitative information about the underlying biological specimen. And in order to do that interpretation, we can use artificial intelligence, okay? So everything that I've shown you up until this point was really done in an optics lab, very controlled setting. Everything's perfect, very well controlled. But at some point, we needed to actually do this in real patients. And that was where we developed, we had an imaging system, a prototype, which I was a part of. And we also wanted to make this easier for physicians, clinicians to be able to use. So everything on the left, we sort of already talked about, but the image on the lower right looks like an H&E image, but it is not. That is taking those 2 channels, 2845 and 2930, and actually developing a coloring algorithm that turns this into the same color scheme lookup table that we all know and love, and that's H&E. And this was although it seems fairly mundane to just color images, this was really important for us to get a lot of backing from our pathologists, and also getting us enough confidence to be able to use these techniques in the operating room, right? Ultimately, we need somebody to do something with these images, and we were able to develop this. So once we had this, we then kind of decided, well, let's show this to pathologists. Let's show this to the real, professionals, and see how they do looking at conventional H&E images versus our own virtually colored H&E images. And when you do that and you ask them to make specific diagnoses, it turns out that the concordance, right, the Cohen's kappa, so the agreement between the two is quite high. So this particular statistic accounts for random agreement. So if you do that, anything really above 85 or 90 is near perfect concordance. So we were able to show that for multiple different diagnostic tasks. And these were shown to pathologists in random order, and they weren't told which is which. They were just asked to make diagnosis based on this. So this was part of the clinical validation. But oh yeah, go ahead. Yeah, so it was kind of a two different things. One is there's a clinical diagnosis that comes from the clinician pathologist who's reading them at the time of surgery. So we have that, but in addition to that, we also have the final diagnosis, which is different. That's where we have paraffin embedded images and slice them really nicely. And then we can do all the immunohistochemistry or in situ hybridization sequencing that we need to get the real final diagnosis. So we also have that as a sort of ground truth, if you will. And it's a good point though, because pathologists are wrong about this intraoperatively, right? That's part of the problem. That we know historically we have some values on this, that their accuracy rate, best guess is somewhere about 90%. The problem is that that's not 90% for getting this final diagnosis, where this is for sure, this is a WHO grade 2, IDH mutant glioma or something. Is this a glioma or not? Is this a metastasis or not, right? So it's these very coarse diagnoses that, defining an accuracy is hard, but I'd say 90% or so. Yeah. So anyways, that's how we got the ground truth. And this was our really first introduction into, I guess, what you can call deep learning. At this point, this was pre-TensorFlow, actually. So we had some people in lab who had some familiarity with some feature extraction software. So this is where you're computing low-level image statistics. And in this case, they use what's called WinCharm, which is fully automated. You can put in any image and you're going to get a fixed dimensional feature vector, 2048 or something. And you can just use that as features for a multi-layer perceptron in this case. So this is just a normal feed-forward neural network. And at this point, we had enough patients and enough sort of tumor classes to be able to train this and validate it. And that's what we were able to do. Similar as what I was mentioning before, we had these really coarse diagnoses. So NL on the right-hand side, sorry, I guess, does this work? No, that's okay. Okay, no problem. So this is normal here. This is low-grade glioma, high-grade glioma, and non-glial, right? And this is where, sure, okay, so if you have these really broad categories, maybe there's something to it. It's somewhat helpful. But again, this doesn't get nearly to the granularity that you need for actually really good decision support in the operating room for how best to operate on these patients. So this was our first go at it, really a feasibility study that we could do this larger scale computer vision based decision support. And then this is where I really came in and started developing my own ideas and my own techniques for this. And one of the things that we knew we needed, the original study in Nature Biomedical Engineering was focused on adults. And I thought, well, why don't we do this in pediatrics? One, because it's a lot more important, because you get a lot of these lesions in this area called the posterior fossa, which is where the cerebellum lives. And they all can look very similar, and they all similarly have really different prognoses, but also have different surgical goals. And I thought, okay, well, we have a fixed set of diagnoses that can happen for these patients. Is there a way to kind of tailor a specific classifier for this task using our images? So I kind of went back to basics, and I'm really interested in single cell anything. And I think it's important for any diagnostic system, particularly for computer vision, because we know that computer vision is going to be better at doing single cell analysis than humans are. There's really high frequency features. It's really hard to somehow give a summary statistic for all the different cells in an entire image. And I think that's where computer vision can really shine, when we are looking at single cell features. So I was doing that. We were just doing simple segmentation and then computing some morphology characteristics of the cells. That was true for two different cell types. In this case, one were the tumor cells and the other one were these tumor-associated macrophages, which we know are very predominant, particularly in malignant tumors. So we were able to develop two different pipelines that were able to segment both tumor nuclei as well as tumor-associated macrophages. And again, I'll just emphasize the reason that that's easy for this is because in this lower image down here, see these bright areas? This is actually, these are lipid droplets that these phagocytic macrophages just, they're just, chemotaxing around, they're walking around in this tumor and they're actually phagocytosing these lipid droplets from necrotic cells. So these show up really bright, beautiful. They're full of foamy lipid membrane, right? So this is something that you cannot see effectively on H&E. Again, a major difference between our imaging technology and H&E. So these are just some statistics that we were able to compute. So things like nuclei count, tumor-associated macrophage count, and then the comparison between these two. And then again, here's just some different features, cell morphology features. These are huge numbers. I think this is like 750 million different cells. So there's a lot into these, but, and then similar things. So this is where, I wanted to ramp up what we were using in terms of the prediction and random forest. For those who don't know, is a decision tree based model. You can use these really out of the box and they're actually quite robust. They tend to not overfit much, and you can do simple classification with this. And we were able to do that. And for this, we were just trying to focus on, is this normal brain or is this lesional? Is this low grade or is this high grade? And again, a simple decision point, but that was really important for differentiating these pediatric tumors as to how you need to treat them, whether you need to be aggressive surgically or if you can just do a minimal subtotal resection and back out. And here's our results. We were able to do really well. We really had kind of 100% accuracy, but again, I hate saying that because this is cross-validated data. These are all the patients that we had in our entire data set. But nevertheless, this gave me the idea that we could potentially do this at a much larger scale. And I was excited about this project in particular because it's the first time we'd done anything really with sort of single cell analysis and using that as a way to do prediction. So, okay. I include this slide because I think there's a bit of variability in terms of how familiar people are with a lot of the traditional computer vision, digital image processing, and then deep learning or convolutional neural network-based image classification. So traditional vision, I've kind of been hinting at, it's where you have an image, you want to compute some statistics. There's no learning involved, whether it's edge detection or histogram of gradients or scale invariant, feature transform, and FFT, whatever it may be. But you're going to get some fixed number of scalar values that serve as your features from which you can train whatever classifier you're like, K nearest neighbor, logistic regression, random forest, whatever you want. The learning is happening with that second stage, right? So with logistic regression, whatever it may be, the learning is only happening just on those features, right? You've already got your features. There's nothing interesting that's going to happen there. You fix that as the data scientist. The difference with deep learning and with specifically deep convolutional neural networks is that you allow the model to learn what the best features are. So you're not going to tell it whether it needs to look for an FFT signal, or it needs to determine what the what the number of edges are, or it needs to learn a specific edge detector, like should you use a Sobel edge detector or something like that. All of that should be learned end to end based on the data. Now, one of the problems with this, as a lot of people know, is it does take a lot of data to train these. These are very over-parameterized neural networks, and there's always the presumed risk of overfitting. In order to get this done, you want to have a sufficient amount of data to train these well. People have been coming up more and more with ways to alleviate the burden of doing of needing a lot of data. But this is the gist, and this is the main sort of core difference between what I think of as sort of traditional or classical computer vision versus deep neural networks. The other thing I want to point out is there's nothing bad or inferior at all about conventional computer vision, right? There's a lot of very good applications for all of these things. It's not like one is better than the other, or you're not cool if you're not using deep learning. None of that is true. It's just, this is the difference in techniques. This is a great slide that I stole from Jan LeCun, who invented convolutional neural networks. But this is the idea, is that for the top row, traditional pattern recognition, you have some feature extractor that's non-learned and a trainable classifier. There's some like feature extractor, then mid-level features. This is basically SVMs. And then you have a trainable classifier on top. And then deep learning is where you're basically training it end to end, and you can train all of these parameters throughout the network. So this is my introduction to convolutions here. So this is what's happening in convolutional neural networks. Those of you who don't know, so a convolution is really just it's a numerical operation that's defined using a matrix. And usually the matrix is small. It's just like, 3x3 or 5x5. That's what's used in the conventional neural networks. And this is an example of what would be an edge detector. So anytime this filter is going to look over every single receptive field in the image, it's just going to march all the way along, and it's going to compute the inner product between this filter 1 in this case, this edge detector, and the underlying receptive field, and it's going to give you a scalar back. That's sometimes called the activation, especially if you put it through a nonlinearity. And you can orient these any way you want, of course, like normal edge detectors. And if you march this over a block M, you get this feature map here where, sure enough, the edges are highlighted, edges that are bright on the left and dark on the right. And then if you throw away all the negative values, you get this map over here where everything 0 is dark and all the things that are bright are yellow. So this is a nice edge detector here. You can do it in the opposite direction and you get these other highlighted edges, or you can do it on the diagonal. All that looks great. You can do it on different images and you get different results, right? You're going to see different edges depending on the underlying image. But then if you look at this in biological tissues, you kind of stop and you think that looks a lot different than this, where you have this beautiful, nice solid line, right? Everything looks fantastic. The problem here is it all looks noisy. I don't know. There's not that much information to me using a linear edge detector for biological tissues. It's probably important, but I don't know that. Why would I use this feature, this specific edge detector, to do classification? I don't have any reason to believe this is any better than just a random filter, right? There's no way to know that. So the idea is don't define it. Don't hand engineer it. Let that be learned. So that's what convolutional neural networks do. And there was a big turning point, big breakthrough in 2012, where AlexNet was submitted to the ImageNet challenge, which is now a big image classification challenge. Now it's gotten so good that, I don't know, it's kind of a joke because we beat humans now, and the error is, it's hard to know what the label is for some of these images. But the point being is that there was a huge jump in the overall performance of these networks when we submitted the first generation of convolutional neural networks versus a lot of the older computer vision stuff. So I encourage you all to go back and read this paper. It's sort of a landmark paper in computer vision. Since that time, everything has gotten bigger and deeper and more parameters. And the models tend to get better, especially with more data. We know that depth is important. We know that in general, the deeper the networks, the better the performance. And people have tried to figure out ways to build these networks better and better. So, okay, now that we've gone through all that, can we use a convolutional neural net to do much more robust and granular brain tumor diagnosis? And it turns out we can, and that's what we did a couple of years ago. And the general workflow is what I've mentioned: is patient comes in with a brain tumor, has an MRI, we do surgery, we get a surgical specimen, it gets loaded into our microscope that lives in our operating room. We acquire an image. These are large images, about 3 by 3 millimeters, sometimes 5 by 5 millimeters. And then we just actually patch these. So you can densely sample from these. You can pick your step size and you generate the number of patches that you like. We do a little bit of image processing, and these images are, again, raw images. We don't use the H&E because those are post-processed images. And then from there, we're able to feed that into a neural network that we can train based on these patches. And then we also developed an inference algorithm that we can use that basically sorts these into diagnostic and non-diagnostic, diagnostic and normal brain. We obviously just want to focus on the diagnostic tumor regions. And then from there, we can just have an output, a probability distribution over the classes that we are interested in for brain tumor diagnosis. So the way this looks in terms of output, so everyone is on the same page, is you define some set of diagnoses. We did the 13 most common brain tumor types. And the output is just a probability distribution over those diagnoses. So you just have a high probability density where the network thinks this is a likely metastasis or likely glioblastoma, whatever it may be. And to do this work well, I'm a big believer in this, and part of my lab is really about anybody can publish AI papers on anything. You can have an Excel spreadsheet and you can write an AI paper. The problem is that in order to actually make a difference is that you have to do some very robust validation and testing to demonstrate that this is going to generalize well. And I believe that the only way to do that is one of two ways. You either need to do this completely prospectively, potentially blindly, or you need to have a prospective and completely external multi-center data set. It is held away from you. It is not yours. Some other institution is actually going to keep that data away, and they give it to you, and then you predict on it, right? So that's what we did. We had a multi-center prospective trial that we used after I had done everything that I could to optimize the network, doing all the cross-validation. We then had three centers, and we compared it, our imaging technique versus our imaging technique with our model versus humans with conventional frozen sectioning, and see who wins, right? So these are some of the results. This should always be in any trial where it shows you what's been included and what's been excluded and why it's been excluded. And then also these are the centers that we included: Columbia, Miami, and Michigan. And these are the results. So on the left-hand side is conventional diagnosis. On the right-hand side is stimulated Raman histology and a convolutional neural network diagnosis. And we designed this as a non-inferiority trial. That's important because that determines your sample size. So we were able to design it as that, just to show is it as good as conventional diagnosis, and it was. So we were able to meet our primary endpoint, and we looked at two different metrics. This was part of the recommendation from our initial set of reviewers. They say you need an overall accuracy, which in this case, we beat the pathologist, but what's important is that the pathologist actually did a little bit better for some of the rare classes. So that means that the mean class accuracy, so that's how well you do averaged over each class, they do a little bit better as well. And you can see one of the interesting things about this, it's not exactly clear from this, but there was no overlaps in the error. None. And that's really important. This is where I think this kind of work really shines, because it really demonstrates how we can use these techniques to augment what humans do. We can actually improve human diagnosis by using these methods. It's not competitive in any way. So if you have a network that's going to predict well on the specific tumors where humans are going to predict poorly, that is a good thing for everybody. So this is an example of how that could potentially help with this. So an important step for me and feedback that we had gotten, which I agree with entirely, is that if you're going to do conventional image classification in a clinical setting, what happens when you feed a tumor in that the network has never seen? I've never seen this diagnosis. It's a rare diagnosis. I don't know what to do with this. It's going to give it a diagnosis that's definitely going to be wrong, right? Because it's never seen it. So one of the things we wanted to develop was a score that would tell you what's the likelihood this has been seen in the past versus whether it's something well within the distribution of images that we've seen prior in the training data set. So I was able to develop this Mahalanobis distance-based confidence score. And really all it is, it's we call class conditional, meaning that you're going to minimize this quadratic form of the Mahalanobis distance, where f of x is the image that's gone through the network F. And you're going to subtract that from the specific class mean. This is the mean vector for let's say glioblastoma, whatever specific brain tumor class you have. So you're going to get a vector out here. This is the precision matrix, the inverse of the covariance matrix. And then again, this is the quadratic form of that. So it's the same term here. And you can compute this for any tumor, anything that goes in. And if it's large enough, you can actually set a threshold to say, if it's larger than this specific scalar value, I don't want to predict on it. It's something we haven't seen in the past. So, sorry guys, I think I maybe, oh, sorry, no, oh, it's here. So again, so when you compute these scores, blue are tumors that we've seen. So these are classes, tumors that live within the training set, and the red are tumors that don't. So things like choroid plexus papilloma, clavichordoma, these are all rare diagnoses that we see in neurosurgery, but not frequently. And you can see if you project this onto the first and second principal component, you get a large variance away from the center. These are computed along the layers of the neural network. You can see red far away from the origin is bad. And then if you project this into what's really important, which is the linear discriminant axis, that's where if you draw the decision boundary for values greater than or less than 0, do you separate these two different groups, meaning trial tumors or common tumors that we included versus rare tumors. And we were able to develop a linear decision boundary, which is great. So one of the knocks on deep neural networks is that they're black boxes, right? They're uninterpretable. I don't know what's going on in them. It's especially bad for any diagnostic system because it's important for clinicians to trust the prediction, right? Why is it predicting this? In the absence of that, sometimes I think it really hinders people's belief in this work. And I think it's our job for, myself and people in my lab and people working in this area to sort of dispel that and try to do what we can to make it somewhat interpretable. So one of the things that we did was what's called activation maximization. This has been published in the past, but the idea is that take your trained network. So the weights are fixed. In this case, theta here, pick a spot in the network, preferably somewhere deep, because sometimes it's quite a bit easier to understand what's happening. And what you want is, rather than optimizing over the parameters theta in the network, you actually want to optimize over the image X. So here, H is the location in the network. This is again, the network, the function that you are computing. L is the layer, J is the specific filter in there. So you can imagine you're way deep in the network, 159 layers in. I'm picking filter 12. All right, way down there. Now I want you to give me the image that's going to maximally activate that specific neuron, really. I want to see that firing down, way down in the network. And I want to optimize the image for that. That's the argmax part here. Our contribution was that this X subject to Rx means that we actually invented this regularization term that helps make the images look better. And if you do that, you actually get these really cool feature maps. You get these really cool activation images here. So this is like, for example, filter 12. This is filter 101. This is 148, et cetera. And you can see that it actually has learned to identify specific features in the images. It's learned to see, is this a small cell? Is this a large cell? Is there complex chromatin? Is there axons? So this one I especially like, this one got a lot of reaction, is that it's actually looking for what we were mentioning before, myelinated axons, which was, a big find. There's no reason for this. There's no reason for you to believe that this is necessarily going to happen. When you do the same experiments for any natural vision problem, for example, if you're going to train this on ImageNet, it looks like noise. I encourage you to look up this technique because it does not look well at all. We have this in the supplemental figure in our paper, but if you do this for natural vision, we still don't quite understand what these feature maps look like or these activation maps look like, but ours is different. Because this is an iterative process, you can make these cute little GIFs. I'm not sure this helps anything, but you're looking at the network slowly optimizing the image as a gradient ascent steps in the image space. Usually you're doing gradient descent steps in your parameter space for your network, but this is the opposite, where you're slowly developing from a random image that's just Gaussian noise. You're going to go to something that maximally activates some specific convolution within your network. You can look at these throughout the network. You can do this at any arbitrary position, and if you do that, you basically get more increasingly complex patterns, which is good. There's this hierarchical composition to deep convolutional neural networks. There's a belief that that's how the brain works, the human visual system works as well. But this is sort of what you get as you march up deeper into the network. You can quantify this. I don't want to go into this too long because we're running out of time, but you can quantify what is being activated based on different tumor types. And when you do that, these are the same filters. This is the equivalent to what I was showing you before with the block M and the convolutions and what I was mentioning earlier, except this is for the learned convolutions here. So filter 148 and layer 159. If you develop those same feature maps, you see, for example, where this specific filter 101 really picks up these large cells with complex chromatin structure. Same thing here, you get these axons. This is what it's looking for. And if you look in gray matter, which makes sense because gray matter is normal brain, you're going to have myelinated axons there. You also get similar bright activation. So when talking about convolutional neural networks, the convolution part really refers to this feature extraction part. This is where you have to go from an image, variable size, whatever it may be, down to some fixed feature vector, right? That fixed feature vector is really what holds the semantic content of the image. What are the relevant things to look for to do some task, whether it's object detection or classification, semantic segmentation, whatever it may, you're gonna write a caption from that image. All of that information must be contained at some point in a single feature vector, 512, 2000, whatever it's going to be. And that's where the difference between what I think of vision and something like NLP or classification or an MLP. And the important thing to look for is that once you've finished that visual feature extraction, how do we make sense of it? What's going on in there? And in particular, with respect to the task that we gave it. And in this case, it was image classification. So if you do a dimensionality reduction technique called T-distributed stochastic neighbor embedding, that just takes high dimensional data and projects it into the plane, you get this. And I intentionally color it this way because it's an unsupervised technique. It doesn't know anything about brain tumor labels or what classes or any of those things. All it really tries to do is say, if in this high dimensional space, if there's a high probability that this specific point is going to be my close neighbor, then I want it to be a close neighbor in this low dimensional space. That's really what the whole algorithm does. And when you do that, you see, sure enough, that the same tumors tend to cluster together, which is what you would expect. This is what you want, right? This means that similar tumors look similar when you get done doing all the visual processing. And the cool bit about it too is that you actually see some of the similar tumor types cluster together. So gliomas here, this is glial up here, particularly pilocytic astrocytomas, diffuse low-grade gliomas, glioblastomas, ependymomas, that's all in the glial category. Non-glial tumors tend to cluster pituitary, meningioma, and then interesting small round blue cell tumors. Don't worry about what that means, but it just means that they all look alike. CNS lymphoma as well as medulloblastoma also cluster together here too. So basically we're doing what we want to do. We're extracting the right features. They may be. Yeah, there are different subtypes. Excellent point. Yeah, so we're actually working on that experiment. So there's very well-defined molecular subtypes for medulloblastoma that we know are different tumors. It's really not one disease. And one of the things that's important is one of the subgroups in particular, we know surgically has a different goal. So we've been thinking about trying to do that specific thing that you mentioned. So great point. You can do, because we're doing this patch-based classification, you get semantic segmentation basically for free because you're densely sampling over the whole image. And the good thing about it is that if your step size for your patch does not exactly match your receptive field, you can actually, you can improve the probability. There's a smoothing that you can do based on pooling the probability for each one of the sort of the patching regions that you're using. So again, as you go through, you're going to get sort of a probability density, and there's this averaging that's happening for each location in your image, and you can average over that, not based on some simple averaging like median filter or Gaussian blur or something like that, but it's actually where there's a probability that's being output by the network, and you can average that probability based on the prediction of the specific neighboring patch region. So this gives us these nice smooth looking heat maps, and this is great. This is really important when you're going to interpret these as humans. You want to know if it's wrong, where it's wrong, and why it's wrong. So the other bit that's really advantageous is that particularly if you're at the margin of a tumor, you can say, are we at the margin and where is it, right? Is it all this stuff normal brain versus tumor? And here you can see this red is tumor and green is normal or gliotic brain. We're able to delineate this margin really effectively and really help for how close we are to getting to normal brain when we're resecting these tumors. You can do the same thing with metastasis. Even when the brain looks normal, you can have these metastatic rests of tumor, which can be important. These are all microscopic tumor infiltration. And then the extension of this was that there's embedded within this big problem is a set of really hard problems. And one of that is differentiating recurrent tumors from what we call pseudoprogression, which is when you have a tumor and you're treating it with radiation or chemotherapy, there's a tendency for those tumors to end up looking, or I should say the adjacent brain to end up looking really angry. It will enhance, it will get inflamed, there'll be edema. And sometimes it's true recurrent tumor, and that's bad. You want to know that versus, oh, maybe it's just pseudoprogression. And it turns out that humans have a really hard time with that. And it's because the actual treatment makes the underlying tissue look abnormal, even when it's not. It's basically just reacting to all of this treatment effect. So they actually have tried to develop strict guidelines to help clinicians determine whether this is recurrence versus pseudoprogression. And it's still very much an open question. They're all basically equally bad. We've tried some things, they've been retracted. Some things don't get reassured, don't get reimbursed by insurance, et cetera. The important bit is that one of the most definitive ways to do it is if you have a histologic diagnosis. And the difficult thing again is this is the heat map for pseudoprogression and recurrence and non-diagnostic. But the thing I want you to see from this is these images are large and they're very busy. There's just a lot going on. There's blood, there's necrosis, hyalinated blood vessels. There's just a lot of gliotic tissue. So it makes this problem really hard. And we wanted to attack this specifically because it's clinically relevant, but also humans really struggle with it. This is the algorithm that I developed for specifically for this problem, because this is different than what we're showing before. Before, you just get a discrete probability distribution over the classes, just pick your argmax, whatever it's going to be. What is the most probable diagnosis? That's your diagnosis. It's a little bit more complicated than that, but that's the gist. This is not like that. The problem is that they're likely going to predict some probability that this is tumor, right? Because it looks bad. This is bad tissue, and we don't have, with all of this, we don't have single patch level labels. These are all weak labels. We have a whole patient or a whole specimen diagnosis. But the problem is that you have to set some threshold. You have to say, if it's above this probability for tumor recurrence, then I'm going to tell you this is recurrence. But that threshold is not known a priori. That's going to be a function of your data set, but also the underlying model that you've trained, right? The annotations that you have. All of those things are something that you don't necessarily know a priori. So we basically developed this method that allows us to threshold this diagnostic recurrence based on pooling the probability distributions and also throwing away regions that we think are non-diagnostic. We can, if anybody has questions on that later, we can talk about it. So I was able to define what we thought was an optimal threshold at 30%. This was based on a cross-validated data set that we used. Again, these are less common. Pseudoprogression particularly is less common. But I really tried to max out what we could with the data that we had, did a lot of rigorous cross-validation to find this optimal tuning. And then this is what you get. So again, it's much more mixed, and nothing really looks normal. The treatment effect doesn't look normal, and then you can have a mixed recurrence as well as treatment effect, and then you can have dense tumor as well. But in the end, we did this external validation. We had Columbia had this data set. They were doing something completely different. They were doing RNA-seq on recurrent tumors. So we were able to benefit from that. And we had 50 patients, and we were able to do really well, 96% accuracy. And in particular, what I liked is the sensitivity was 100%, because it's important as a clinician, you want to think about what's the important things to not do. How do I not hurt somebody, right? That in a way is a difference between accuracy and sensitivity or any other metric. In this case, you certainly don't want to miss if it's recurrence. If it's recurrence, that's bad. You don't do anything, right? So you want to set your threshold to be on the lower end, so your tool is more sensitive than specific, let's say, for example. So the last bit here is, I think, the initial title of my talk. But in saying all of this, the general momentum of brain tumor diagnosis, and I would say cancer diagnosis in general, is defining tumor types based on molecular features. What that means is, for this specific tumor, is this mutation present or not? Or are these 3 mutations present or not? Or is there some chromosomal aberration? Is there some epigenetic alteration? All of those things are the tendency that the World Health Organization is moving towards to define these very rigorous diagnostic classes. It's true for brain tumors. It's true for lung cancer. It's true for a lot of different things. So, 2016 came along, and sure enough, there was a revision to the brain tumor classification scheme, and they said now intrinsic diffuse gliomas, which are the most common malignant brain tumors, are going to be defined by a set of molecular features, and that's the deal. If you're going to give a real diagnosis, you have to do immunohistochemistry or sequencing, or some test to define this group. So again, the histology, this is where we stopped. We were in the histology land here. So we were able to tell you whether this is an astro, an oligo, or a glioblastoma. But everything south of that line was IDH mutant, is now the molecular feature. So IDH mutant, IDH wild type, ATRX loss, P53, 1P19Q codeletion. So all of these things now are absolutely essential for defining these diagnostic subgroups of diffuse gliomas. So the question is, can we extend a lot of the techniques that we've developed here to the molecular classification of diffuse gliomas? Now, I'll tell you, this is a much harder task, because now you're mapping pixels to tumor genotypes, right? There's no specific reason, and humans aren't able to do this, but how do you know that a specific mutation is going to generate a visual feature difference, right? Because that's what this task is about. Can I map a set of visual features to specific molecular subgroups? And that's the hard part. It's true for medulloblastomas, glioblastomas, whatever it may be. And that is something that is really a superhuman task, meaning that humans currently can't do it. And it's an open question as to whether this can be done. People have tried it on different data sets like MRI, H&E, but there has yet to be something that I think is a real robust, generalizable approach to it. But I can tell you we're working on it, and we have a pathway to, I think, get this done. Certainly, we have used much more, I would say, high-powered technology to do this optical image feature extraction. My PhD student, Chang Zheng, he's here. He's been working on a lot of contrastive learning methods to get this done. And then in addition to that, what I've been trying to do is really develop this multimodal training procedure where we're actually using sequencing data from TCGA, open source. We can actually train a gene embedding model from that. And then the classifier can use the semantic information contained within that genetic embedding layer to be able to do classification on visual features. So there's people have done sort of gene to vec models, but it hasn't been done constructively for visual tasks. It's been done in natural language processing and natural images. People have tried to kind of combine sort of semantic features with visual features, but it's yet to be done robustly in a biomedical data set. So that's kind of what we're working on. And I can just tell you that preliminarily, the data is looking quite good, particularly for some of the most important diagnostic mutations. This IDH, which is isocytosine dehydrogenase, which is by far and away the most important mutation in gliomas. We're improving more and more with the additional data that we get, and also with this gene embedding method that I was mentioning to you earlier. So that's it. In conclusion, SRH is a rapid label-free optical imaging method that provides high-resolution diagnostic images. Digital images of fresh biomedical specimens. And the combination of SRH and deep neural networks can achieve rapid and accurate brain tumor diagnosis. So, oh, sorry, guys. Let me go back. There we go. So this is my group, up and coming, fantastic, energetic group. I run the machine learning and neurosurgery lab. Again, if you're interested in any of this stuff, we are hiring. We have some projects that we have funding for and need help with. So if you're interested at all, please reach out to us. We'd be happy to meet with you. So that's it. I think I got done in time, right? Oh, yeah, good. Any questions, please? Go right ahead. Thanks.
What besides this new application that's
Mm-hmm.
what would be some of the major barriers to getting it into hospitals?
Oh, yes, that's a good question. So I've been involved a little bit in sort of the FDA approval of AI. And I can tell you that people still don't know how to do it. Meaning that I think that there just isn't a good precedent. It's not an attack on the FDA or anything. I think it's really hard to know how to toe this line of what is really useful. I mean, we're all trying to help people. We're all trying to help patients. So we need something that's useful, but we also need something that's not going to put the company at a lot of liability for risk, right? Like if you're an AI system and you make a wrong diagnosis, like so diabetic retinopathy, for example, huge space for computer vision. You don't triage someone to being seen by an ophthalmologist when you should have. What do you do with that now? A patient goes blind or something. It's like, so what happens? And the problem is that a lot of the companies are like, I don't want anything to do with that. We don't want to be liable for hurting anybody. And I think they mean that in earnest. But at the same time, that's what the tool is meant to do, right? So it's not hurt people, I mean, as a triage. So I think it's really challenging to know how to make these products really helpful, but not having the liability fall back on these companies for making diagnostic errors. In the case of this, I think we run into that. The thing I'm interested in these high bars. Like I want to do these really interesting, scientifically complex things. And with the company that we work with, they're like, no, we want the simplest product you can think of. I want to do like, is this tumor or not? They even want to do less than that to say, is this tissue that you can use for diagnosis or not? Which to me is not interesting at all, right? But there's this tension between what they can do feasibly and what they, trying to minimize the amount of risk. And the FDA they also want to minimize the risk of their products, while at the same time making something clinically useful. So I think we're still sort of towing that line. But I think we will get there. It's just a matter of making these models better and learning more and more about how to make them into, usable and feasible clinical systems. Yeah, so. Anybody else? Anybody online?
I just like to congratulate you, Todd, on a tremendous trajectory of work and a very substantial lesson in how long a path it is to document and explore all the variables and come up with a system like this. It's quite impressive, of course.
Yeah, it's definitely been a long road, even though, you have to really enjoy the process. And, it's been a lot of fun. And I think I consider myself fortunate to be part of something that really started really in a lab. I mean, it was on an optics table at a chemistry lab, and to see this through to now where I'm using it in the operating room to treat patients is, I think that's a gift. I think a lot of investigators don't ever get to see that. So, even at this early stage, I consider myself quite fortunate. While there's still
Great.
I don't ask questions really yet. Like, I'm gonna go right over my paper first and then figure the rest out.
Do you have any idea?
Yeah, in vivo, basically. Yeah, so that was something that we have been working on that. So there is a working prototype of an in vivo imager where you'd have a probe, basically it's a handheld probe, and you can use the same sort of fiber laser technology that we do now to capture the image, but it's sort of in this handheld device. I do think that's feasible. I mean, there are ways to do that. One of the major challenges is that we're acquiring these two image channels, and they're temporally separated. You acquire one, the laser shifts, and then you acquire the second one. That in itself is not a problem. Exactly. So it's the movement of the actual, either the brain or your hand. Like, how are you going to hold it still, right? So it's like, do you mount the actual handheld thing using some rigid arm, or do you hold it, or you try to do this extremely robust sort of error correction algorithm? Do you do it on the software side? All of those things I think are good questions to have. But I do think that in the long run, we need to develop methods that are, in vivo, that are non-lethal, that we can image these things, without having to remove tissue, because it's the right observation to have that, let's say you're at the margin of a tumor and you're imaging and it's normal brain, right? You don't want to have to cut that out to tell whether it's normal brain or not. You want to know in the resection cavity. So. Yeah, I think you definitely lose image quality the deeper you go, especially in this epi-reflectance mode, where you're basically shining the laser down and you're trying to collect all the laser that comes back to you in the handheld objective. But I think they've gone something as deep as 80 microns or something. So it's doable. And this is where, again, this is one of the things that I believe in is, again, it comes down to clinical task. If it's for margin delineation, right, that's basically a binary classifier. Is this tumor or not, right? When it's quote unquote simple like that, you might not need your images to be as good, right? You can take some noise and you can still have a fairly robust classifier for that. So that's where I think, understanding what you need out of the data, out of that observation is important, not only for what you build and the actual objective that you're going to use and all those kind of things, but also if you want to classify an AI sort of computer vision decision support model to go along with that, knowing what the expectations are from the data itself. And, if it's a binary classifier, you can maybe make your objective a little less good, or you can take some noise in the data set. So. Yeah. Mm-hmm. Right. There is. Yeah. I'm glad you mentioned that. So we have a whole research program in lab right now based on exactly what you said. And it goes back to the noise question is, so we have very good theoretical models for where the noise comes from. And it has to do with this combination of this additive and multiplicative noise. And we believe it's probably Gaussian distributed, as most noise is. But we know that it's not so simple when you're going to like deconvolve the image, or in this case, we're trying to use image to image networks like UNet to try to denoise these images. Now we know that we've tried this. If you just use simple Gaussian noise, it doesn't work. So you can't just say, oh, we're going to noise these images to train a UNet to be able to denoise them using some conventional loss function like mean squared error or something like that. For whatever reason, you can, on your training set, beautiful, looks great, perfectly denoised, right? But then when you actually take a new image that's just come out of a patient, it does not work. So we have been trying to use exactly what you said, where let's use what we know about the optics of stimulated Raman scattering microscopy to determine what is the best noise vector, right, that we can use to try to deconvolve this sort of inverse image problem. So we have been working on that, and it's important. I don't think you can do, we're doing image classification, but I think if you want to improve image quality, you have to understand the optics of the system for sure. Great, excellent. Thank you all.